Super. I think we're going to go ahead and get started. My name is Marc Penn. I'm a cardiologist by training, trained at the Cleveland Clinic, spent 25 years there, but have a deep expertise in inflammation and clinical markers of inflammation. Started a company called Cleveland Heart Lab that used inflammatory biomarkers to define cardiovascular risk. Sold that to Quest Diagnostics, and I'm a part-time medical director at Quest. I also serve as the chief medical officer of Keystone Bio, where we look at the relationship between periodontal disease, and specifically Porphyromonas gingivalis as a driver of periodontal disease, and its cause effect on Alzheimer's disease. We know, as I'll go through, that there are multiple connections between gum disease and systemic disease, not only heart disease, Alzheimer's disease, metabolic syndrome, things of that nature. And being able to eliminate Porphyromonas gingivalis and eliminate every one of its mechanisms of inflammation, I find very compelling and interesting. Keystone Bio is a clinical stage biopharma company where we have a unique precision antibody that removes Porphyromonas gingivalis from the mouth. I think it's key to recognize that you need to have a precision antibody or a precision approach for the mouth. You do not want to eliminate all bacteria from the mouth. The mouth generates most of the nitrates in the body and it maintains vascular tone. If you remove all bacteria, that's actually a negative. We have strong IP portfolio around this precision therapeutic, and we have a good team in place focused on Alzheimer's disease. So again, there's a robust literature linking periodontal disease to systemic disease, and so much so that about 20 years ago, a group in the UK, using an antibody developed in Zurich, looked at whether they could remove Porphyromonas gingivalis from the mouth. So if you look at the panel all the way to the left, these are patients who all have periodontal disease, and you see the vast majority of them have Porphyromonas gingivalis in their mouth. The before being treated with this antibody, cleaning of the mouth removed Porphyromonas gingivalis, or at least removed measurable Porphyromonas gingivalis. The reality is in the sulci of the teeth, the bug still is present. Those in green received dosing of the antibody, KB001, around each of the teeth. Those in blue received saline. At 6 months, you'll see the folks who received saline, in green, they had, almost all of them had recurrence of the Porphyromonas gingivalis. So less than, as you know, we normally go to the dentist every 6 months. Less than the time we normally wait, the Porphyromonas gingivalis comes back. But in those patients who received the antibody in blue, it did not return. And in fact, it didn't start to return and returned slowly out to a year, whereas virtually everybody who received saline had PG in their mouth by the end of a year. So this is evidence that we can use a precision antibody to actually remove bacteria from the mouth. And we were starting to develop that as a treatment for Alzheimer's. And again, why are we looking at an antibody that was developed 20 some odd years ago? It's because of the growing literature that links Porphyromonas gingivalis in the mouth to the development of Alzheimer's disease. There are multiple mechanisms, and key multiple mechanisms, that Porphyromonas gingivalis can use to drive inflammation and drive Alzheimer's disease. Outer membrane vesicles are released by the Porphyromonas gingivalis in the mouth. The bacteria does not need to get into the body to have a significant impact on systemic health. These outer membrane vesicles get released into the bloodstream. They contain lipids, lipoproteins, mRNAs, and multiple different things, including gingipains and endotoxin, LPS, from Porphyromonas gingivalis, all of which, when these outer membrane vesicles land, can induce local inflammation at the site. And again, the bacteria don't need to get in, so the blood-brain barrier isn't inhibiting the delivery of these outer membrane vesicles to the brain. If we look at gingival tissue and stain them for PG with our antibody, you see that it's there in the gingival tissue. It's in the choroid plexus of patients with Alzheimer's disease. It's also in the frontal lobe. And again, this is in the absence of any bacteria in the brain. These OMVs really are the toxic mediator of the presence of Porphyromonas gingivalis in the mouth. Our antibody targets the heme gluten target within these OMVs that are necessary for the bacteria to survive, so it becomes a toxic antibody to the bacteria in the mouth. Again, there's a very large literature that supports the link between Porphyromonas gingivalis and Alzheimer's disease. And one of the mechanisms by which it affects is decreasing the decreasing the biofilm that these bacteria create in the mouth, which again, when the dentist is able to do scaling in the mouth, they remove some of the biofilm, but they aren't able to remove it all, which is why it comes back so quickly. These data here show that the antibody and gold particles on the right in the image actually bind to the bacteria, and the biofilm of these bacteria actually are significantly reduced. And by reducing the biofilm, the body's native immunity is able to actually then remove these bacteria from the mouth. The GAIN trial focused on an inhibitor, COR388, that inhibits gingipanes, a single specific pathway that Porphyromonas gingivalis uses to induce inflammation, really showed the potential of this approach, right? They did a dose-finding study in patients who had mild to moderate Alzheimer's disease, patients who all had periodontal disease, and about a third of them had active Porphyromonas gingivalis in the mouth. When they looked at the patients who had Porphyromonas gingivalis active infection, they actually had about a 60 to 70% reduction of PG in the mouth. So we're unable to eliminate it just inhibiting a single pathway. But importantly, they showed a 50% reduction in cognitive decline in this mild to moderate population, which is a really significant finding from our point of view, but also importantly shows the potential of targeting Porphyromonas gingivalis to treat Alzheimer's. While the GAIN trial had a significant effect with a published 57% reduction in cognitive decline, the question we have is how much more can we achieve by blocking all the pathways that Porphyromonas gingivalis induces? Importantly, it's been shown in a number of studies that NLRP3 inflammasomes are a significant activator in Alzheimer's disease. They also play a significant role in multiple diseases, including rheumatoid arthritis, heart disease, atherosclerosis, and the such, which makes sense given that PG is linked to multiple disease states. These data here show that gingipanes, looking at KDP136 mutations in the bacteria or KDP117, both of which block gingipane either generation or release, that they're still able to induce NLRP3. So this paper and others have demonstrated that NLRP3, again, a key mediator of inflammation in the inflammasome, actually is not inhibited by inhibiting gingipanes, and we believe that by eliminating the bacteria using our approach, we have a better chance of eliminating all the potential mechanisms of PG and having a greater effect. So while the GAIN trial, we believe, was really important, really well done, and really showed the path forward for the field, we think that KB001 precision monoclonal antibody is important. It was safe in a phase 1 clinical trial. It neutralizes PG and all the targets of PG, eliminates 100% of PG at least for 6 months, which we believe is the key because, again, patients go back to the dentist every 6 months. It's simple companion diagnostic we're developing, and it's a topical treatment. There is really very limited systemic exposure. And it has a downstream potential for prevention. Where new chemical entities often can't be used for prevention, we believe that keeping the mouth clean is critical. Our phase 1 study, using a humanized form of this antibody that we've developed and optimized for use in humans so that the Porphyromonas gingivalis itself doesn't cleave the antibody and render it inactive. We'll be repeating the study that was done with the UK compound, where we'll give subgingival injections around the teeth in patients who are PG positive, who are greater than 50 years of age and stable, not necessarily with Alzheimer's disease. We'll give 1 or 4 doses to patients. The original BOOST study looked at 4 doses over 10 days. And we'll quantify Porphyromonas gingivalis in the mouth, again, with a primary endpoint of 6 months clearance. Phase 2, which will commence immediately after that, once we have dosing clearly demonstrated, as well as an additional safety study, we'll look at patients who have evidence of mild to moderate Alzheimer's disease. We will dose based on what we learned from the phase 1, and we will look for changes in cognitive function and biomarkers associated with Alzheimer's. It's important to note that inflammation is not specific. It can induce multiple disease states, including Parkinson's disease. And there's clear evidence that Porphyromonas gingivalis can drive Parkinson's disease. And we're excited about broadening out our focus beyond Alzheimer's to, again, Parkinson's. There's an interesting study that shows that dental scaling decreases the risk of Parkinson's disease. So if you take patients who have never had periodontal disease, presumably not having Porphyromonas gingivalis, and they continue to go to the dentist and get routine scaling over 5 years, they have a much lower incidence of developing Parkinson's disease compared to those who have a history of periodontal disease and periodontal disease who still go to the dentist. Because again, even if you go to the dentist, if you cannot clear your Porphyromonas gingivalis, your risk of developing these systemic inflammation and systemic downstream diseases still remain elevated. So in summary, we believe the GAIN trial, an important trial, really validated PG as a target in Alzheimer's disease with mild to moderate cognitive decline. It showed benefit of this approach is restricted to those with active PG in the mouth, which is, again, is our goal is to eliminate that active PG. And it revealed that the population of Alzheimer's disease patients who are positive for PG antibody, is 33%. We're not slicing out a small portion of Alzheimer's disease. This is actually a significant percentage, and I'm sure no surprise to anybody in this audience. There's multiple causes of Alzheimer's disease, and we're going to need to identify the right patients for the right therapies. In relation to our approach and the magnitude of benefit in relatively later stage patients, we think it shows the benefit of decreasing inflammation in the microenvironment of the brain. We've shown in other disease, and my lab's shown in cardiovascular disease, decreasing inflammation restores function of hibernating cells. There can be neurons that are active that are just dysfunctional because there's inflammation. By preventing them from going on to further death due to ongoing inflammation, we believe we can restore function. It prevents further cell death, and it enhances microvascular function and allows scar remodeling that can lead to significant restoration of tissue function and certainly prevention of further decline. It also suggests, I think importantly, that removing PG will likely be a component or a companion of future other therapies. If you want to have an antibody that's going to remove beta amyloid, you also want to eliminate the inflammation that's driving that beta amyloid. So if there's an easy way to quell inflammation and then attack the amyloid deposits, likely to have a better benefit, and maybe the combination will bring amyloid antibodies into the mild to moderate or more severely affected patients who now really don't get benefit, and we need to treat people very early. Finally, we are ready to go on to our phase 1 study. We finalized the shift in the focus to the humanized monoclonal antibody again. And as I mentioned, this was really required detailed work understanding the antibody and making sure that that antibody had limited immunity, but also was not subject to cleaving by the Porphyromonas gingivalis itself. We completed the design of that. We rendered it non-cleavable. We demonstrated improved target binding characteristics with the humanized compared to the murine, and we finalized the formulation. So we're ready to go, and we're ready for IND-enabling studies with a goal to be done in 2024. So I want to thank you for your time, and happy to answer any questions, but we're really excited about this space. We think targeting inflammation is key, and we think for our approach, targeting all the pathways for Porphyromonas gingivalis is important, and while it'll have impact in Alzheimer's, we believe will have impact in other disease states as well. Thank you. Thank you.